Our COVID rapid tests, how do they work and what is the science behind them? This is a review article on protein-based lateral flow assays for COVID-19 detection. So first, if you recall your early science classes, capillary action is how they work. So if we take something like this and connect it with water, you see how the water travels up. Water is sticky and likes to travel, and the great thing is that liquids like water can carry molecules as it travels across a membrane. So the basic components of a lateral flow assay, our rapid tests, are antigens, possible viral RNA, so that's from COVID, our proteins, our biomarkers, we have the cartridge itself, and then we have the swab saliva and the serum. So for the specifics, the lateral flow assay, the sample pad is usually made up of cellulose. And this cellulose is loaded with a labeled detector protein. The test pad itself is usually made of nitrocellulose, and this is loaded with the immobilized antibodies to the sample protein or the detector protein. And at the end, there's a wicking pad to absorb excess sample. So visually, the sample gets loaded at the beginning. You have a conjugate pad, which has the targeted antibodies, which would then attach to the COVID itself. And then you have the virus test line and the control line with the wicking pad at the end. So the sample is loaded onto the sample pad. So this is what you do when you collect your nasal swab and you load it into that liquid when you're mixing it in. That's going to be dropped 3 or 4 drops onto the sample pad itself, which will then flow to the conjugate pad, which has the primary antibodies that'll attach to the COVID. After the COVID and antibodies combine, they continue flowing along, and these are now called conjugates, and these conjugates will then interact with the test strips that have been sort of plated into 2 different distinct rows. And the first row will have antibodies which will attach to this combined COVID antibody. So if that passes along, it'll attach to the first one, and the first line will connect these and hold them in place, and it'll trigger a fluoroscopic reaction to make that pink marker. But if they keep flowing and they don't get they don't react, they don't get triggered by COVID being present, then the antibody itself being travel across will react with the second one because there's a second line that attaches primarily to just the antibody. So again, visually, you have COVID, it's going to flow along, it's going to interact with these. If there is COVID in the sample, then these will continue flowing along. And as these are flowing along, the COVID samples with the antibody will attach to these antigen markers here, causing that pink to show up on the test line. If there are no COVID, they'll continue along, and you'll see like that yellow circle. The yellow circle is now attaching to this other plated antibody, which is the control line. So this one is targeted with COVID, this one is targeted to just the antibody itself, and that's how you get the distinction between the two. So how do we get these antibodies to begin with for these tests? Well, these are monoclonal antibodies that are derived from mouse hybridomas, which will then produce specific antibodies in large quantities. Well, what does this mean? Essentially, we infect a large quantity of mice with the virus. It goes through a natural immune response, producing a lot of antibodies to COVID itself, and then we essentially drain the blood from these mice, extract the antibodies they produce, and then we use those for our rapid antigen tests. This is how it's been done, and this is just what happens. So literally, all we have to do to update our rapid tests is to take current viral strains and inject these mouse with the antibodies, and then use these antibodies that the mice produce to update our rapid tests so that they're more specific to current strains. And why do we use this way? Why do we do it this way? It's because these monoclonal antibodies that are produced by the mice have a high binding affinity and specificity to the target itself. So these monoclonal antibodies have a high specificity. That means that they're not interacting with other things. They're interacting largely just with the COVID virus itself. But the sensitivity is where we're having issues. So not having enough virus would be a lower sensitivity. Basically, there's not enough virus to cause the reaction that you expect to occur. So in March 2021, we had an average sensitivity around 90%. Where in a previous paper we looked at when the viral load is less than 10 to the 6 copies per milliliter, the wild type to Omicron variants decrease. And then when you have sufficient viral load, it's a very clear linear decrease from wild type to Omicron. And these rapid tests, these rapid antigen tests, the rats, are designed for the wild type. So they become less and less sensitive to about a coin flip, and probably less by now since we're on XBB, meaning that these tests are well overdue for needing to be updated. Going back to the paper, also of note, while viral load is measured by viral RNA copies per milliliter, this is highest during the infectious period, which is ranging between 2 days after exposure, so this is before symptoms, to 12 days after showing symptoms. So a major limitation of these rapid antigen tests are that they're only indicative of a person having COVID, not if the person is capable of transmitting COVID to others or not.